We continue now with our study of the immune system. This is the second in the series of 2. And we have transitioned now to the acquired immunity. So in the first presentation, we discussed everything in general as an introduction, and then up through the innate immune response, or the nonspecific response. And we reflected on the transition points between nonspecific and specific, such as the antigen-presenting cells. And now we begin to consider how we acquire a specific response to an invader. So an acquired immune response reacts to and targets a specific foreign molecule called an antigen. So there's a lot more detail about antigens, and I encourage you to investigate it a bit because it's very important for understanding this complex means by which we can generate a specific response, and also to understand why you have to be injected, get a flu vaccine every year. The viruses have diverse populations of genes for those particular proteins, the antigens, and the antigens you're protected against this year might not be the same ones that are on the viruses that are trying to attack us next year. So it's important to learn more about antigens and in the context of how the immune system protects us. So the presence of a foreign antigen activates several types of lymphocytes to combat an infection. So there is active immunity, which occurs when the lymphocytes are exposed to foreign antigens in the body. Then there's passive immunity, which occurs when we receive antibodies made elsewhere. So requires another organism. There is natural passive immunity, such as breastfeeding, and then of course there's clinical practice of being injected with an anti-venom if you were bitten by a snake or a spider or something like that. So cytokines regulate both innate and acquired immune responses and also help coordinate these responses. So each lymphocyte clone has a unique surface receptor, and all the lymphocytes contain proteins on their surface capable of binding a foreign antigen. So the lymphocytes that can bind a particular antigen are called a clone. So a clone is a group of lymphocytes that are specific to one antigen. So you see the little semicircular, and then the square, half a square, and then the Y shape, or half a diamond, right? So it's representing that there's a particular antigen or antigenic determinant. I mean, this is at the point now where it's down to the 3 to 8 or whatever amino acids in a protein, or the particular conformation of some other macromolecule, biological, or toxin or something like that. So, as I said, the study of antigens and how they work together. In particular, you should look up hapten, H-A-P-T-E-N, to understand how very small molecules can generate immune responses. So you can become allergic to penicillin, for example, right? And that's because you're exposed to it once, and now there's cells that have a very specific, a clone that has a protein surface marker that is specific just for penicillin. Okay, so this clone would be maybe generated against something else, and this clone against something else. So if you've ever heard of the term of monoclonal response, this would be a monoclonal response. But if you are injected with a protein, you might produce what's called a polyclonal response, where even though these are each just different little parts of one particular protein, like the H1N1 proteins that you were inoculated against, there's different parts of the H1N1 proteins. In other words, antigen-presenting cells could break them down into multiple types of antigens, antigenic determinants, and so you can produce a polyclonal response to the same H1N1 protein category for those viruses, and it gives you a broader spectrum of protection. Whereas if you're just getting a monoclonal response, it's quite limited, because that antigenic determinant can be different from year to year. that particular part of the amino acid sequence. The mutated form of the virus just puts a different amino acid in there, and it might be a little more complex than that. But anyway, and it alters the antigenic determinant that this clone was generated against. It no longer works against that flu virus, right? So this obviously is a very cursory introduction to this, and I just want to try to get you to appreciate it from this point, thinking of antigen, antigenic determinant, hapten, H-A-P-T-E-N, and then monoclonal versus polyclonal responses. So these are things that you should do an internet search on and read just a little bit more and build upon your appreciation of this superficial introduction to it. The types of lymphocytes are B lymphocytes and T lymphocytes, and the NK cells. So we said that the NK cells also reflected this transition from non-specific to specific. Certainly the B lymphocytes and the T lymphocytes are well within the specific category, but remember that the T helper cell, I think we had mentioned, and we'll see it, interfaces with the antigen presenting cells. So some subsets of these at least will provide the receiver of the information at this transition from the non-specific to the specific. So in other words, the transition depends upon effective communication. The steps in the immune response then, a naive lymphocyte lymphocytes have not yet been stimulated. Another term that's used for these are virgin lymphocytes. You may find that in other more advanced readings or slightly older literature. The antigen stimulates a specific lymphocyte clone of cells that have surface receptors for the antigen. The clonal expansion occurs as the cells multiply rapidly, that should be a Y there, in response to the antigenic stimulation. Right? So the decision now has been made that we have a line of protection in place from a prior exposure to this. So in the text they state that the assumption, this description is based upon the assumption that there was a prior exposure to this antigen. So some new cells become one of three types of effector cells: the plasma cells, the helper T cells, and the cytotoxic T cells. So let's assume that you had the swine flu this year once, and now you're exposed to it again. You got around somebody else that had it, and now new exposure has occurred, and this is how your body's going to respond. Say, man, after the two weeks of suffering that we went through, we're prepared for this second attack, and we're going to beat it before it gets into us. So the other cells become memory cells, which create a stronger and more rapid response to future antigen exposure. So second exposure can contribute to strengthening the reserves, as a way to think about it. And as we proceed through the illustrations, I'll try to build upon that. So the first time you're exposed to antigens, you set up a population of memory cells. So it's like next time they come, we've got this reserve to be called into force as quickly as possible. But it's a limited size because it's a single exposure. Boom, you get exposed again. Your memory cells are going to divide and make more memory cells. And so we've all heard of booster shots, and that's in effect part of what's going on, as we will see. So let's keep that in mind as we proceed so we can appreciate this expansion. So clonal expansion leads to the immunologic memory. So we have, we're envisioning a single, although there's going to be a whole population of them, throughout you that each is exposed to the antigen. It's some new invading antigen. We want to determine whether it was there before or not. We're working on the assumption that it was. So Here we're considering a primary immune response. The antigen is binding to a naive lymphocyte. So we'll back up. We now have reversed our assumption. It is not a re-exposure. This is a new exposure. So we went through the sampling and we determined, this is a new antigen. So naive lymphocytes will reproduce. So there's a resistant strain of the H1N1, and it persists to next year. So now next flu season, you get exposed, and it's seen as a completely new antigen. Like, well, what? This isn't H1N1 that we saw last year. This is a new one. So it starts a new response. So it can be a re-exposure, but it's a re-exposure to one that now has antigens on its surface that you do not recognize, so you're not protected against it yet. So this, again, is why you need to get flu shot every year, because there's such a wide variety of flu strains, virus strains, that when we remove one from the global population, others become more common and more prominent, and then they're the strain that's going to get us next year because we're not protected against it. So this would be the case. This antigen could even be viewed as the H1N1 vaccine now. If you didn't get the swine flu this year, say you just got injected with this. So it is viewed as new, and the naive lymphocytes are going to reproduce. And as they are reproducing, they are, you notice that they produce this half-moon marker on the surface, and through this initial clonal expansion you get short-lived effector cells that carry out the immediate response, but through this clonal expansion there is a subset of cells that are going to just kind of nestle aside somewhere within the lymphoid tissues distributed throughout the body, lymph nodes or whatever, and create memory cells that are long-lived and continue to reproduce. In other words, they sustain mitotic capabilities. Then with the second response, say the other one was the vaccine that you got, and now you, after your an appropriate time after your vaccine, you go to a classroom and there's somebody spreading H1N1 virus all over, and you get exposed again. So the memory cells respond quickly and more strongly to the this follow-up antigen exposure. So the clone expands more rapidly to create additional effector and memory cells, and quite often, although memory cells do distribute throughout the body, they will be at their highest density, population density, at the initial site. Say we're thinking about it coming through the respiratory and oral tracts, the nose and the mouth, right? And the lymphoid tissues in the mucosa there, or in the tonsils, as we had mentioned, or even down into the larynx and the trachea, the upper respiratory tract down into the lower respiratory tract. This is where the memory cells certainly will be located, but they will also distribute throughout the body. So in other words, at the site of initial entry, you would expect that to be the site of any repeat entry, as long as it's a traditional route of entry. Natural, let's say, not so much traditional. So now we have the re-exposure, and then it's a potent trigger of mitotic activity, and more memory cells are going to be produced. So from these new sets of cells, some are going to become memory cells, and some will become the active cells. So if these are B lymphocytes, they'll contribute to the humoral immunity. The aka B cells, or B lymphocytes, are responsible for this. The clonal expansion of the B cells results in the production of the plasma cells. So here we have to remind ourselves of a reserve cell, a memory cell dividing to reproduce itself, more memory cells, and then other cells, daughter cells, that are going to differentiate into, even terminally differentiate into the final functional cells that will live their life, do their job, and then they have an endpoint. They don't live forever. But these memory cells are, we hope anyway, going to live as long as we do. There's some uncertainty as to how long they may live. If you were vaccinated at birth, are those memory cells still with you when you're 80? So we're learning more all the time as we ask these kind of questions. So those that are determined to terminally differentiate and do their job become the plasma cells. And then each clone of plasma cells produces a specific antigen-binding protein called an antibody. So again, this humoral-mediated response should occur at the point of entry of the re-exposure. So in other words, in the oral and nasal cavity, you want some memory cells to the H1N1 if you had the swine flu or got vaccinated. If you had it before and that was the point of entry, then you should expect that there'd be a really high number of these memory B lymphocytes ready to divide and produce plasma cells and gear up the antibodies, so that as the virus is coming in and beginning to divide in that area or reproduce and spread, you've got plenty of antibody to stick to them and inactivate them and prevent the further spread, and also trigger their destruction by other means, and also to contribute to the destruction of the cells that are infected. So the memory cells are also produced, allowing for the rapid and stronger secondary antibody response if the antigen is encountered again. So we're thinking of booster shots, but also the multiple exposures. Think of how many times you might get exposed to the swine flu virus throughout a whole season. Even just thinking of how it spreads through a family. One person gets it, and then another person when that person's getting better, and by the time you're fully better, if you were the first one, the third member or fourth member in the family now has it, and you're fully re-exposed after completing your cycle. So we're all aware of these kind of things. So again, we're thinking about this humoral-mediated immunity or immune response, and the memory effect in a humoral response is illustrated by these two slides. We see the primary response with an antibody concentration. Then they're looking at the antibody concentration in the plasma with the first antigen exposure, and in time over weeks. So here's about 4 weeks. And then the antibody concentration in plasma. So I just recently had to get the hepatitis vaccination because I work in a clinical setting occasionally, as do all people that work in healthcare facilities. There are certain immunizations that you are required to get. And so the first time I got it produced a response like this, and about 3 or 4 weeks later, they called me back in. I got the second one, and so now I should have produced a very large response that should protect me from that virus. And of course, I'll always test positive for the virus in a test that would look at antibodies, because I now have the antibodies in my plasma. But if they had a test to look for the viruses, they'd see that I don't have a virus load, but I do have the antibody, and then that means that I just was exposed to it before, and then I show them my immunization record and go, oh, okay, you got immunized. So you don't have this viral infection, this hepatitis disease. You are protected from it. So now let's consider the antibody as a chemical itself. You can see that there are 2 heavy chains and 2 light chains, so it is a hetero-tetramer, right? Hetero because they're different. This might be the same as this, and the light chain the same, but the light chain and the heavy chains are not the same, so it's hetero, and it's 1, 2, 3, 4 proteins. So a monomer is a single one, remember, and then this is a tetramer. So it is a hetero-tetramer. And so each antibody has 2 antigen binding sites, and the light chain along with the portion of the heavy chain that is has the disulfide bond holding them together, forms what's called the FAB region. And at the remote end of the FAB region is the antigen binding site. Here's where you're going to see the highest variability. It's called the hypervariable region because it has to be adapted. The sequence of the amino acids here has to be adapted to have the specificity to match whatever antigenic determinant this single clone of antibody was produced against, right? And then there's a hinge region where the molecule is actually bent, and at that point is where we see 1, 2, 3, 4 disulfide bonds holding together the two heavy chains. And so from that hinge region to the other remote, the opposite remote end, from the opposite from the antigen binding site, is called the FC fragment. This is constant. So the antigen binding, that's the FAB region, and the FC, this is constant. And this is the portion, the FC region. This free end of the molecule is the part that binds to the antibody receptors on the WBCs that express those antibody binding antibody receptors, right? Antibody binding receptors. So the antibodies are also known as gamma globulins, a particular type of globular protein produced in the body that were classified as the gamma category. So there's obviously an alpha and a beta, and now a gamma. Here we see the gamma globulins. So there's different types of globular proteins, different classifications. The antibodies fall into the gamma globulin. There are 5 classes of the gamma globulin antibodies based upon the FC region being different. So it's constant, but constant relative to the antigen binding region. Right? So so there's variable, there's a constant region, variable, and then hypervariable as you proceed toward the antigen binding end of it. But there are different classes of these constant regions, different genes producing a set of them, right? So there's a set of FC amino acid sequences for the FC regions that are called IgG, or IgA, or IgE, or IgM, or IgD. I usually put the M there and say gamed, G-A-M-E-D, but anyway, whatever helps you remember it. So the IgG is the most common. It's the secondary response. So if you receive passive immunization, typically it's the IgG that you would be receiving. The IgA is a secretory form, so it's found in saliva, tears, and breast milk and other areas, typically the mucous membranes. And then the IgE is involved in allergic responses. The IgM is the antibody involved in primary response. And the IgD is found on the surface of B cells. And the role of the IgD is still unclear. And it's been unclear throughout my complete set of years of studying biology. So I'm not sure if the researchers just can't figure out what it's doing, or there isn't enough interest and money to sustain active investigation. I'd have to look in the literature to find out. So we are then beginning to think about the function of antibodies, of course, with a great deal of attention paid to the hypervariable region for the antigen binding site and the constant region for the antibody binding site if there is a receptor binding to the cells that we'll see here, and we're going to go through this in a set of slides. So number one, the antigen binding to the antibody at the antigen binding site will activate B lymphocytes if the antibodies are already bound to receptors on the surface of them. Now remember, we earlier looked at the macrophages having antibody receptors expressed on their surface.
but they did not bind the antibodies until after the antibodies had bound to the pathogen. That was to trigger phagocytosis. Here we're talking about signaling a B lymphocyte that is not a phagocyte. It's a secretory cell, right? All it's going to do is secrete antibodies. So this is acting actually as a signal to this cell saying, you have bound an antigen that is either new, if it's a naive B lymphocyte, or one that you were made to fight against, right? So you're a memory cell. So you'll go on to divide and make new memory cells if it's a naive exposure, or more memory cells if it's a booster shot, right? And of course you generate the plasma cells to produce antibodies. So that's the humoral-mediated response summarized that we just considered. Or they can act as opsonins. So we talked about this before. They bind to the structures, and then the antibodies will bind to the receptors on the phagocytic cells to enhance the phagocytosis of the invading organism. Or they cause antigen clumping and an activation of the bacterial toxins. So here's a bacterial toxin. So remember we said that the antigenic determinant can be a single small molecule, like a bacterial toxin. Quite often these are lipopolysaccharides, or LPS, and they can do bad things, so we need to bind them up and inactivate them. So that's still a larger and more complex biological macromolecule than penicillin, but still there's that capability to produce a specific antibody response to individual chemicals versus proteins or glycoproteins on the surface of the organisms, or microbes. I'm trying to recall if viruses are even included in microbes, are they? But anyway, microbes and viruses. Then the fourth function is to activate antibody-dependent cellular activity, such as the NK cell, or in the eosinophils. Remember we talked about the eosinophil if it was a parasite, but still it depends upon an antibody binding to the surface of the parasite. The fifth triggers mast cell degranulation. So in this case, they're still showing it kind of like a bacteria, but it could be a pollen type to which you are allergic. So you would have IgE antibodies on your mast cells, and IgEs can bind the pollen, and when you get exposed to the pollen, then it signals the mast cells in your nasal cavity to degranulate. Their mast cells are loaded with these secretory granules, and they'll just undergo this tremendous exocytosis and dump. Well, if you've ever had a pretty robust allergic response to pollen, you know what I'm saying, right? Just all of a sudden you can feel, I end up sneezing about 1,000 times, and of course go racing for my Astelin antihistamine nasal spray and spray that in, and it does a pretty good job at terminating my sneezing fit. And then after the sneezing fit, it's almost like I can feel the histamine just spread from my nose to my ears to the back of my head, and just inflammation ensues. So this can be a very tremendous thing, and this of course is related to the anaphylaxis, the IgE-dependent massive allergic response that can cause severe problems. And then the antibodies contribute to the activation of complement, and of course the complement can be involved in signaling or other means of contributing to the immune response. So you are encouraged to watch this animation or to read further in other sources. Now let's consider the cell-mediated immunity, the role of the T cells. So in cell-mediated immunity, the T cells are specialized to defend against intracellular pathogens. So everything that we've kind of looked at so far is something in the interstitial fluid space or on the surface of the mucous membranes or the skin or things like that. Now we're thinking of something that invades to get inside the cells. And it's not just viruses that do this. There are some bacteria that actually invade cells and then hide out inside the cell. I think there might be some funguses that do that as well. And so typically we hear intracellular pathogens, we think only of viruses, but there are some prokaryotic and possibly eukaryotic, if there are funguses that get inside cells, that can produce these problems. And so as we're continuing to proceed in this category of intracellular pathogens, let's not just think about the H1N1 viruses or, rhinoviruses that cause the typical head colds or flus that we are familiar with. Think of warts on the skin or genital warts, and hopefully you're remembering the human papillomavirus, right, that causes the genital warts. So it extends again beyond just the HIV immunodeficiency virus that we hear so much about infecting and getting inside cells, or the flu viruses. There are many examples of the intracellular pathogens when you start to think about it. Malaria is a parasite that gets inside the cells, right? So if you start to think about it, this goes way beyond just the simple thing, oh yeah, HIV and H1N1 swine flu. So the T cell receptors recognize foreign antigens only with the major histocompatibility proteins. So the MHC proteins are found on all of our body cells, those with the nuclei, and they vary from one person to another. The reason that we say the nucleated body cells is because we'll see with the red blood cells, these are not present, and neither is the nucleus. So we'll emphasize that when we get to that point. So activation of T lymphocytes. So the cell binds to the T lymphocyte. So remember, this is a macrophage, or the antigen-presenting cell with the MHC receptor. So remember that picture from the first presentation. So after phagocytosis, that ends up presenting the antigen on the surface with the MHC. And then the T lymphocytes are able to recognize that the MHC is there, and then probe the, if you will, probe the surrounding area and say, oh, you're presenting something new and I need to react to it. And so it is a signal transduction that occurs, and this activates the T lymphocyte. So this is called the T cell receptor. The major histocompatibility complex is considered separately on this slide. So these are proteins that help our immune system defend against intracellular pathogens and cancers. So this is how we recognize self. And there are two classes, MHC1 and MHC2. The MHC1 is found on all the nucleated cells of the body. It's recognized by cytotoxic T cells as self, right, or as not self if it doesn't match self. Then the MHC2 are found on antigen-presenting cells only, and they are recognized by the helper T cells. So again, the MHC class 1 is on all nucleated cells. MHC2 is also found in nucleated cells, but those nucleated cells are strictly in the category of the antigen-presenting cells. So there are only three that we had mentioned: the macrophage, T cell, the NK cell, and the dendritic. Are you recalling that? I'm flipping back to that table in my book. So the antigen-presenting cells was the dendritic cell, the lymphocyte, and the macrophage. Okay, so, and then the T helper cell, as we saw, is the one that will recognize that. So when we are thinking of the immune system being in the process of targeting an invader, we think of, MHC2 for the antigen-presenting cells. But for cells in general, for tissue rejection, other than blood, we think of MHC1, other than red blood cells, right? So the T lymphocytes, the development of the effector cells, are considered in this slide. So there's a multipotent stem cell in the bone marrow. So when we studied blood, we considered this. And the lineage to the T cell precursor. And then there's the question mark as to whether that branches to the natural killer cells. And of course, their task is to kill antibody-coated cells, and they are picking up the antigen signal to tell them what to respond to from the MHC class 1 target cells. And then, of course, the T cell precursor migrates to the thymus gland when we are young, and undergoes a differentiation there to become cytotoxic T cells. They are also killers. And then the helper T cells also develop, and what they do, as we saw, is they bind to the MHC2 antigen-presenting cells and secrete cytokines that activate other immune cells. So we're getting the immediate defense, the direct defense, and then the recruitment from the helpers, right? So they help coordinate or orchestrate the immune response. So the functional roles of cytotoxic and helper T cells are considered on this slide. So the cytotoxic T cells attack and destroy the infected cells in two ways. They release perforin to create holes in the infected cells, or they stimulate the apoptosis, that's cell suicide, the gene expression dependent. It's pretty complex and highly regulated. You want it to be. You don't want to have this simple trigger to go, oop, the cell died. You want to trigger it through complex signaling mechanisms so that you've got all of these different checkpoints to make sure that this is really what you want to do. The removal of cells has to be tightly regulated such that the rate of replacement is not exceeded. So the helper T cells, secrete the cytokines that influence other immune cells and bind to B cells, promoting the differentiation to the plasma cells. Again, you are referred to an animation and additional readings. In particular, I'm always referring you back to the textbook because there's information there that you will find on your performance assessments, exams and quizzes and exercises and case studies as a specific resource to build the breadth of your understanding of these systems. The slide presentations are the introductory level to the more detailed studies even within our textbook. And again, these animations, I've watched them and they are really excellent. So if you have the opportunity, and certainly if I require this in a course, you'll be prompted to use them specifically. So common characteristics of B and T lymphocytes is another feature that you should consider. So the immune response pathways are considered on this slide. So there's a general pattern in the immune system responses to infections and antigenic challenges. The innate responses act quickly. Their acquired responses take longer but are stronger, a more robust protective response. And again, this is how you see you're getting sick, you get sick, and then you stabilize a bit, and then you begin to recover. You can think of it that way as well. It's not always just to protect you the next time. The acquired responses that are kicking in with your first exposure are contributing to your recovery. The cytokines from both innate and acquired immune events serve to coordinate and reinforce the two sets of responses. So here we'll look at this immune response to an extracellular bacteria that enters through a break in the barrier, the skin or mucous membrane. So first, the bacteria enter the extracellular fluid from the outside. So it would be the mucosa or submucosa, if it is a mucous membrane, or it would be the dermis or a subcutaneous tissue, depending upon how deep the puncture is with the skin. So the bacteria will enter, and this is, of course, the extracellular fluid compartment, so from the external environment to the internal environment. So what we see then is the bacteria going to activate the complement proteins that are ever present in their inactive form. They will make the membrane attack complex that is going to contribute to the lysis process of the bacteria. And also they will activate mast cells, as we saw, contribute to that. Even if there is no binding to IgE, the complement proteins can signal mast cells to secrete chemotaxins and histamine to increase the permeability. The chemotaxins, as we know, are going to contribute to the attraction of circulating leukocytes, and typically it's a range of things. It helps the endothelial cells realize that they need to put proteins on their surface to make the circulating leukocytes stick to them. Anyway, there's a complex process of generating this infiltration of the tissue, or extravasation, extra outside of vascularization, the process of getting out of the vasculature. The extravasation process is quite complex with a lot of signaling, but basically the chemotaxins contribute to that. And as they are coming in, and once in here, they are following a concentration gradient to the source of the chemotaxins. So again, they're showing one mast cell, but you know that there's many of them spread around here. And so there's a tremendous source of concentration gradient of these chemotaxins. Again, these reduced illustrations can be a bit misleading at times, so I try to expound on that as we are always seeing them. And then the plasma proteins will act as antibodies or acute phase proteins. So remember, the increased permeability allows the leakage of some plasma proteins. So there is definitely a change in the endothelial cells so that they become leaky. So you know that the part of this process is the change, the localized change in the endothelial cells that contribute to their communication with the blood leukocytes, and also their ability to leak molecules into the interstitial fluid that they would not normally allow to leak. The antibodies and the acute phase proteins and the complement proteins act as opsonins, and so you know that there's of the gamma globulin component of our plasma, there's a lot of antibodies present. And so they can take a sample of your blood and see if you've ever been exposed to something before by testing to see if you have antibodies. So we know that there's a lot of antibodies in our plasma, and they are recruited into an area of invasion to contribute at this initial stage to the opsins. And if you've been exposed before, can contribute to a specific part of this response rather than just generally as opsonins to coat and inactivate. And then, of course, the ingestion and disabling or inactivation also, just the destruction by the phagocytes taking place as the I wonder why they have plasma cells there. There we go. That label was there before. So if you're pulling in a monocyte, it can become a macrophage. But remember, the PMN infiltration, the polymorphonuclear infiltration of a tissue area like this is very prominent. So macrophages also would be coming in, monocytes to become macrophages, but the polymorphonuclear or neutrophil response is pronounced, very pronounced. And of course, we're leading to the point of accumulating leukocytes and debris, so pus will begin to develop in this area as part of the process in some cases, as you know. So the plasma cells would, so if the B lymphocytes were not present, they might come in from the plasma if they're there, and then you'd have division in the production of plasma cells. If memory cells were there locally, then they would be activated, as we had mentioned. But now we're just thinking of this exposure, of course, getting to the phagocytes contributing to, as well as antigen presenting to the T helper cells, contributing to the activation of the B lymphocytes to become the plasma cells and secrete more antibodies, however specific, so that they're more effective at contributing to this opsonization and inactivation process. Now let's differentiate that with an immune response to a viral infection. So the virus invades the host, and there's the pre-existing antibodies. So then the surface proteins required for binding to the host cells are occupied, covered, masked, so that they cannot bind to the proteins on the surface of the host cells. That is a critical part of the infection process, so you are protected. This would be the case of a prior exposure or immunization. If, however, some of the virus that is invading has different proteins on its surface, so even though you got the immunization or exposed to a different strain before, it is like brand new, and then it will infect host cells, and some of the viral proteins will be expressed with the MHC1 on the surface, and so the infected host cell will have viral antigens exposed on their surface. Remember, the MHC1 is on every nucleated cell in the body, so this could be whatever virus infects whatever cell. If it happens to be a virus that would infect an antigen-presenting cell, then it would be with the MHC2 as well. But what we're talking about here is not antigen presenting, but part of the process of the viral infection leads to the viral antigens being exposed on the surface with the MHC1. So it changes the characteristic, the surface characteristic of the infected cells is the way to think about it as part of the infection process. It's not an antigen presenting mechanism that is specifically restricted to what's happening with the antigen presenting that is going on here at number 3. So with number 2, macrophage ingests the virus, and here is the MHC2 with the viral antigen as part of the viral antigen presentation process to trigger a specific response. Also, cytokines will be secreted. Interferon alpha activates an antiviral response in the infected cells to try to minimize the replication and the production of virus particles that can spread. Cytokines also contribute to the inflammatory response. Then here, number 3, we get the antigen presentation to the T helper cell that activates the B lymphocytes, and viruses can be present, binding there, and they're saying, oh yeah, we have a memory to this, or we are naive, and this is what we're going to respond to, so let's structure our genes such as the hypervariable region, the antigen binding region of the antibody we're going to produce is going to be to this particular part of the virus, and it becomes plasma cells and secretes antibodies that contribute back here. Then also the T helper cell contributes to the activation of the cytotoxic T cell that has T cell receptors. The T cell receptors will bind to the altered MHC1 on the surface of the infected cells, and this will signal to the cytotoxic T cells to secrete the perforins and the granzymes. And then here, where we finally see the appearance of a cell that undergoes apoptosis and dies. So it destroys its own DNA in the nucleus and breaks down some of the structures and triggers the cytoskeleton to I'm trying to think of the term, to break up the cytoplasm into these little packets. They're called apoptotic bodies. And then phagocytes can simply take these up and destroy it. So they destroy the infecting agent along with the infected cell. But nothing gets spilled out. There's no autolysis and leaking of the lysosomes such that the lysosomal enzymes will dissolve everything inside the cell and rupture the membrane and cause the spillage, because if that happens, then you trigger an inflammatory response. So this keeps everything right in little packets and inactivates all the mechanisms required by the virus to continue to reproduce. And then these little packets, these apoptotic bodies, can be devoured by the phagocytes and removed. Remember, the macrophages or the phagocytic cells are responsible for removing dead and dying cells too. The allergic response is kind of a different category, but similar types of or common mechanisms, but carried out through different chemical signaling and extremes. Of course, it is a response to something that is typically not threatening. I mean, who's ever heard of a pollen infection, right? You have a pollen allergy, but you never get infected with a pollen and grow a cedar tree inside your body or something, So anyway, an allergy is an inflammatory response to a non-pathogenic antigen called an allergen. So it's still an antigen in that sense of having an antigenic determinant that is specific and can be bound by the antibody against that antigenic determinant. But because it is non-pathogenic, we use this term allergen. So there's an immediate hypersensitivity reaction that occurs within minutes. It's mediated by the antibodies that are already present to that allergen. Or a delayed hypersensitivity reaction over several days. It's mediated by the helper T cells and the macrophages. So the same cells, but just check here. Yeah, doesn't do much. But slight variations to trigger different events. And a different class of antibodies. This is the IgE, ultimately an IgE dependent. So here we have the allergic responses. With the first exposure, there's the allergen. Looks like a pollen. The allergen is ingested and processed by the antigen-presenting cells. The antigen-presenting cell activates the helper T cell. And here we see the helper T cell again with the T cell receptor, and then it activates the B lymphocytes. And again, the pollen is present here, the allergen. Could be, if you're allergic to dust mites, like I am. I'm not so allergic anymore because I've been getting shots for years. It's actually a protein in their poop that you're allergic to. Isn't that sweet? Anyway, and if it's cats, it's chunks of their dead skin, the cat dander, as it's called. So the be stuck here on the B lymphocytes, and that's how they either recognize it, if it's a memory cell, or change their DNA so that the proteins that they produce, the antibodies, will be specific for that, and thereby sensitize you to the allergen in that sense. And they will become, of course, plasma cells to secrete antibodies. And typically in this case, it's producing the IgE antibodies. And then memory B and T cells retain memory of exposure to the allergen, and that leads to activated T cells. So these antibodies are the IgE class, as I said. So somehow this process of allergens being non-pathogenic triggers only an IgE response rather than the IgM and IgG of a classical response. And therefore, when you get your allergy shots, what it's supposed to do is desensitize by shifting away from an IgE response to the more classical IgM, and then ultimately with your maintenance shots, your IgG response. So you have IgGs around to bind these up and prevent them from sticking to IgEs and triggering the allergy. It helps, but it doesn't cure. So the IgE on the mast cell, decranulation, as we said, cytokines, histamine, etc., vasodilation, bronchodilation, increased vascular permeability contributing to inflammation, and then the allergen bound to the IgG produces an activation of the complement proteins contributing here as well. And then the activated T cells will release cytokines and contribute to the inflammation. Now we can transition over to talk about the blood cells, so the blood transfusion reactions. So the red blood cells in humans are not nucleated, and they do not have the MHC proteins. They don't they don't have the nucleus to have the genes to express them, right? But anyway, the red blood cells have a shorter lifespan, and they get all the proteins and messenger RNA they need to live their life before they lose their nucleus. So the red blood cells don't have the MHC proteins, but other RBC antigens can be recognized as foreign. So even though they don't have the MHC proteins expressed on their surface, they have other proteins, glycoproteins, and these have been categorized as either A or B. So there are two genes for these, and they produce proteins that act as antigens to recognize as self as far as red blood cells are concerned. And so you can so there's 4 blood types that are possible. You can be, and these are dominant, each of these are a dominant allele. So if you have an A and the other, which doesn't produce any protein, the O, anyway, you can look in the text. It can be AA or AO, BB or BO. Remember, we have 2 alleles for every gene. One from mom and one from dad. Or you can get an A from mom or dad, or a B from mom and dad, and you're AB. Or you get neither of those and don't produce any surface proteins, so it's O. So what you might recall is that the O is the universal donor, especially the O negative for the RH factor and other surface protein. And AB is the universal recipient. So in other words, if you are, well, let's look at the antibodies in the plasma that are not made against self, but to what is absent. Okay, so in this case, we're going to make antibodies to the allele and its protein, or the protein of the allele that is absent. So if you are type A, you have the A antigen. You don't produce antibodies against that because that's you, that's self. What you do produce is antibodies against the other protein allele, so I'll call it, anti-B antibodies. So if you are blood type A, you will have anti-B antibodies in your plasma. So if you are transfused with B type, you're going to have a reaction, right? If you are B, you have the B antigen, and you produce antibodies against the A. If you're type AB, you have both A and B, so you're not going to produce any antibodies, so therefore it's traditionally called the universal recipient. You can get A or B or O, or AB blood, right? As long as the RH factor matches too. The type O blood, there are no antigens present, so you produce anti-A and anti-B, so you cannot receive A or B type or AB type. You can only receive O type blood. But because it has no antigens on the surface, any antibodies, such as anti-A or anti-B, will not react to it. So it's considered the universal donor. I'm not so sure that that terminology is used anymore because we're so cautious about blood transfusions, because if there's a transfusion reaction, the negative outcomes are really quite significant. It's a high-risk situation. But for the purposes of understanding this, that's typically a way to talk about the AB as the universal recipient, doesn't have any antibodies to A or B, and O as the universal donor. Doesn't have any surface antigens for antibodies to bind to. So if you're blood type O, you don't have either of them, and the antibodies in the plasma are going to be against both A and B. If you are blood type A, you have A antigens, you're going to produce antibody B. And blood type B, you have anti-A. If you're AB, you're not going to produce either antibody. So you think of blood type O, you have 0 antigens, but all the antibodies. Blood type AB, you have both antigens and no antibodies. So is the way to think of it. So if you give this type of blood, B, to a type A person, the anti-B antibody's there, and it's going to bind. So a mixture of type O and blood type A is shown here, where you have both the anti-A and the anti-B antibodies, and somebody with blood type O, and you have given them type A, so the type A antibodies in the plasma are going to bind to the red blood cells that were the donor red blood cells that were transfused. And this leads to clumping and hemolysis and a lot of problems. The textbook covers it briefly, but it's more complex. A major thing is that, as you know, these are packets of hemoglobin proteins. And if a lot of these cells are broken, if a lot of hemolysis occurs, then there's a lot of hemoglobin in the blood. It's not supposed to be in the blood. It sticks to the structures of the kidney, and it can cause renal failure. And that's just one part of many bad responses to a transfusion reaction. So the autoimmune diseases, then, the immune system doesn't normally react against self-antigens in a process called self-tolerance. So, we grow up around ourselves, we develop embryologically and fetally around ourselves. So as our immune system develops, it comes to know us, right? Kind of a generic way to talk about it. But there can be errors in some cases, so these would be autoimmune diseases when the self-tolerance system fails. Autoimmune diseases are usually restricted to specific tissues or organs. So we've talked about type 1 diabetes, and let's just look at the list here. Some of the common autoimmune diseases in humans, so consider the disease on the left here, and then the antibodies targets on the right. So we've discussed Graves' disease, hyperthyroidism, where the TSH receptor is the target of the antibodies, and when the antibody binds the receptors on the thyroid cells, it acts just like the TSH and stimulates the thyroid gland. The insulin-dependent diabetes mellitus, type 1 diabetes, the IDDM, or type 1 diabetes, the pancreatic beta cell antigens are the target of the antibodies. With MS, the myelin of the CNS neurons is the target. Myasthenia gravis, the acetylcholine receptor of the motor end plate. In this case, the antibody blocks the receptor, and so the acetylcholine cannot bind to it, and so there's weakening of the muscle. The rheumatoid arthritis, there's an antibody against collagen. Systemic lupus erythematosus, or just called lupus. intracellular nucleic acid protein complexes. There's an anti-nuclear antibody involved in this. And then Guillain-Barré syndrome, which is an acute inflammatory demyelinating polyneuropathy, where there's a target of antibodies against the myelin of the peripheral nerves. In many cases, this is treatable and, in essence, reversible or curable, if you want to think of it that way. So it is an inflammatory response to something, maybe viral infections. It's not really clear, but a treatment for this is called plasma diaphoresis. That helps to remove the antibodies. So the diaphoresis is a mechanism for removing substances, in this case particular proteins, from the IgG class of proteins from the plasma. So you're actually filtering the plasma, if you want to think of it that way, to remove the antibodies against the myelin as specifically as possible. In this way you expedite the recovery, and many of these people recover completely, but quite commonly, unfortunately, there's always lingering deficits. So there's obviously similarities between this and this, but this is irreversible and terminal. It cannot, there's no cure for it at present. There are effective treatments, but no cure. This, in effect, is a short-term excuse me, loss of function and paralysis that is treatable, and in some cases fully reversible, or I guess say that's curable, right? Then finally we can consider the role of the nervous system and the endocrine system, other than all the immune hormones, as a complex interaction. So it's an old idea. There's not a lot of data, but the research in this area is quite active. It's still controversial. The three systems use many of the same signal molecules and receptors, so the immune function is altered by many hormones and neuropeptides. As well, many cytokines impact the neuroendocrine function. So the textbook gives a very interesting presentation about the types of hormones secreted by leukocytes, like ACTH and things like that. Definitely referring you to the text to read more on this topic, as we're just going to have a cursory overview of it here. So pathogens on one side and physical and emotional stress on the other. So, of course, they are a brain drain, if you will. And, you know, at the end of final exams, a lot of people get sick. So we're approaching that part of the semester, and we're all familiar with that. So there are neuropeptides from the brain, the nervous system, that can affect immune cells, and from immune cells that can affect the brain. So, you know, when you're sick, you feel like crap, right? Can't concentrate, you can't think, sometimes can't even barely be conscious, right? And then the neuropeptides, of course, are affecting endocrine cells and hormones that will affect the brain. And then hormones from the endocrine cells affect the immune cells, and the cytokines from the immune cells affect the endocrine cells. So hormones, therefore, can have other tissue target cells producing other types of effects, as well as certain protein hormones. The stress alters the immune system function, so the nonspecific stimuli, the stressors, disrupt homeostasis. There are two types of stress that elicit two types of a response. An acute stress results in a fight or flight reaction. Remember, a classic one of the endocrine cell, a type of hormone that affects the immune cells, are the adrenocorticosteroids, cortisol, glucocorticoid also, because it affects the glucose homeostasis. But we also remember that it's a steroidal anti-inflammatory, so there's an inhibition of the immune system by the adrenocorticosteroid. So that's part of the acute, and then there's the chronic stress that elevates the cortisol levels over a period of time, and that suppresses the immune responses. The neuroimmunomodulation and stress is considered in this slide, kind of a concept slide with cellular and molecular studies of the chemical factors related to stress overlapping with descriptive clinical studies of the association between stress and illness, both of which overlap with studies of the systemic responses of stress, and then where all three overlap, you have this common zone that is not well known or understood yet, but there's definitely recognized overlaps and can have significant impact on organisms in their responses to environmental challenges such as invading organisms. So our summary slides then, I'm not going to read through them, but we initially considered up through, in the first one, up through the innate immune response, and then we now have completed our consideration of the acquired immune responses and the interactions between the three control systems.